to host Dr. Bill Banks, our head of research at the VA here. He's made really seminal contributions to understanding the blood-brain barrier and how it facilitates communication between brain and the rest of the body, and in a lot of different contexts, including aging and neurodegeneration, and most recently COVID. So it's really exciting to have him here today. He's a geriatrician and also in the Department of Internal Medicine at the University of Washington. I found out through his CV he trained at Tulane and also did an endocrine fellowship there and started his career at the VA through the New Orleans VA. And then he returned to his home state in Missouri, where he was at the St. Louis VA and St. Louis School of Medicine until 2010, when he moved here to Seattle. And he is currently our ACOS, our Associate Chief of Staff for Research at the VA. And he's had numerous awards, including the Middleton Award, which is the VA's highest honor for researchers nationally in 2018. Anyway, thanks for presenting for us today.
Sure, thank you. I'm delighted to speak today, I really am, and particularly to more of a clinical audience, because in the last 10 or even really in the last 5 years, the area of blood-brain barrier, and particularly the areas that I'm interested in, have really become translational. So my plan for the talk today is to start out with some basic science, what the blood-brain barrier is and how it relates to neuroinflammation. And then after that, to go on and give some examples of clinical diseases that can be explained or involve those processes. And then at the very end, I'll follow up with some of our recent work we've done with SARS-CoV-2 crossing the blood-brain barrier. Oops, there we go. So it's hard to think of a disease these days, particularly one that affects the brain, that doesn't seem to somehow involve neuroinflammation or neuroimmunity. And here I've listed a handful of the ones that I've been interested in or have worked in, but there's almost any disease I think you can think of is now involved in this. And this wasn't the way it always was. Back about 40 or 50 years ago, neurology and immunology, I think, were pretty solidly separate fields. And the blood-brain barrier was the main thing that separated them. And when those rules were violated and somehow neurology got into immunology or vice versa, it was never good. You know, things like multiple sclerosis or something like that. But over the last 50 years or so, these guys have really changed. And now we're much more of in the field of neuroimmunology, almost a yin-yang interaction in much of the CNS areas that these impinge on. The blood-brain barrier is still there, and it still separates, but it also has other functions. This is I'll talk about today. So there's much more of an interface or an integrator, as well as a barrier and separator. And as a result of this, another big change has been that neuroinflammation isn't just for diseases anymore, but is probably even more important in normal functioning, normal physiology. So it's involved not only in pathological sleep, but in physiological sleep as well, not only in insulin resistance, but normal insulin transport, the microbiome, peak glycoprotein regulation, and the normal trafficking of immune cells into and out of the CNS. So all of these areas are sort of new within the last decade or so. About 3 years ago, Shelley Erickson and I were asked to write a review for Pharmacological Reviews, and we decided to write on the blood-brain barrier involvement in a neuroimmune axis. And as we began looking at the field, we realized that that day had long passed, that there was so much information that we had to divide it up in some way. And so we wound up talking about blood-brain barrier involvement in 5 separate neuroimmune axes. And these are somewhat arbitrary divisions and definitions as categorizations often are, but I think they're very useful to think about the panoply in different ways that this field is involved in. So the first one is regulation of tight junction and barrier function. We've long known that inflammation disrupts the blood-brain barrier, but increasingly it's being realized that the normal regulation of tight junctions, what tells the blood-brain barrier to be tight and to be a barrier, uses a language of molecules that we would recognize as being neuroinflammatory or neuroimmune ones. One area that my labs contributed to a great deal is the transport of neuroimmune substances across the blood-brain barrier, in particular the blood-to-brain transport of cytokines and chemokines. We also now know that the barrier cells themselves, the brain endothelial cells and choroid plexus cells that form the CSF blood barrier, secrete cytokines, nitric oxide, prostaglandins, and all kinds of neuroimmune substances. It's almost as if they were immune cells themselves in terms of that aspect of neuroimmunology. Another area is that the ability of immune cells to traffic to the blood-brain barrier. This doesn't occur because the blood-brain barrier is disrupted, but rather it's a highly orchestrated interaction of the blood-brain barrier with the immune cell. And finally, modulation of blood-brain barrier functions. Just as the blood-brain barrier is secreting substances to affect the cells around it, it's also receiving signals, neuroimmune signals, from circulating immune cells, from distant peripheral tissues through the blood, and even from the CNS, the microglia and astrocytes. So it's in constant communication using a neuroimmune language to do various functions. So let's talk a little bit about what the blood-brain barrier is. The discovery is often attributed to Ehrlich, who won the Nobel Prize for things like the magic bullet, basically created pharmacology. And he actually got the interpretation wrong. It was the students that had to straighten this area out. But nonetheless, Ehrlich was the first to do, or one of the first to do this kind of an experiment. Injected a dye, which normally circulated and distributed throughout the animal tissue. Here we've recapitulated this experiment using trypan blue. And many of the tissues stained, in fact, all of the tissue stained, except interestingly, the central nervous system. So if we were to look at the liver or the lung or anything like that, it would be such a dark blue with the amount of dye that we've injected, it would look really black. In fact, here's the inside of the skull, and there's not much here but bone and dura mater, which is outside the blood-brain barrier. And yet here, less than a millimeter away, is the brain, no staining at all, except for in a couple of places like circumventricular organs, but basically the brain remains unstained. And the reason for that is because the capillary bed that goes through the brain is modified. Here's the liver capillary, probably the leakiest capillary bed in the body, and you can see it's really almost a sieve. And it's a sieve for several reasons. Here are these window-like structures. These almost look like cathedral windows. And indeed, the term is fenestra, the Latin for windows. And then there's these big holes, which are various things, but primarily macropinocytosis. And then if you use your imagination, where this liver cell would, endothelial cell would about another liver endothelial cell, they wouldn't really oppose very well, and they would leak between the cells as well as through the cells. Well, the blood-brain barrier gets rid of all three of these things. Wherever the endothelial cells, brain endothelials come together, they're cemented together by tight junctions. You can see this line right here. There's very little pineocytotic activity, and the fenestrae are all gone. So there's this physical barrier in the cell wall, but the blood-brain barrier is more than that. It also maintains barriers by being an efflux system. So there are some brain-to-blood efflux transporters that grab things that are trying to get into the brain, or in some cases are already in the brain, and export them back out into the blood. And it's also an enzymatic barrier, which means that some substances are prevented from getting across by being degraded by the brain endothelial cells. The best known example of this are the monoamines. There's lots of epinephrine and norepinephrine and acetylcholine circulating in the blood. If they could get into the brain, this would create mayhem, and barriers, enzymatic barriers there prevent that from happening, as well as the cell wall barrier. So this is great for the brain on the one hand, because it means it keeps all this circulating riffraff out of the central nervous system and lets it do its business. But it's bad news on the other hand, because that means that there's not a nourishing ultrafiltrate being produced to nourish the brain. So how does the brain get all the vitamins and minerals and amino acids and glucose that it needs if there's no ultrafiltrate? And the answer now, again, is the blood-brain barrier. The blood-brain barrier is endowed with all kinds of transporters for all of those substances, efflux substances to help regulate homeostasis, and even transporters for things like peptides and regulatory proteins that help in brain-to-body communication. And that's an area that I've been most interested in. And things like leptin and ghrelin and some of the gastrointestinal hormones are good examples of how those help to communicate with the brain, the nutritional needs of the body. So how does the blood-brain barrier, the capillary bed, know to be a blood-brain barrier? And how is it knowing how much, let's say, GLUT1 to produce? And the answer is it's in constant communication with other brain cells, and as well as peripheral cells as well. And the communication part with the other brain cells is often referred to as the neurovascular unit. So we have pericytes and mast cells and astrocytes all communicating with the brain and blood-brain barrier and the capillary bed, and sort of informing it of the brain, the needs of the brain. So the blood-brain barrier, in one sense, is a slave to the central nervous system, and it changes throughout health, throughout development. And so it's trying to serve the needs of the changing central nervous system, or central nervous systems whose demands have changed because of disease. And many of the molecules that are involved, as I said before, that are involved in this communication are what we would recognize as neuroimmune agents. So I can't talk about all 5 axes, but I want to talk a little bit about 3 of them and then move on to some CNS diseases. So I'm going to talk about transport of neuroimmune substances across the blood-brain barrier and secretion of neuroimmune substances by the barrier and modulation of barrier functions. So first, transport. So this goes back, our first paper to about 1989, and we were able to do this because of this wonderful mathematical equation by Patlak, Blasberg, and Finstemacher. And basically, the math takes the heavy lifting so the experiment becomes easy. Essentially what we do is we inject, in most cases, radioactive material into the bloodstream. And over time, we take samples of blood and brain, and we use this equation to calculate the unidirectional influx rate into the brain. What this equation really does, interestingly, has very little directly to do with blood-brain barrier. It allows us to integrate the signal in blood over time so that we're able to study substances that are quickly cleared from the blood. And that's really the magic of this equation. Produces data like this, which has several aspects of interpretation, but the one that we're usually interested in is this part, the linear aspect of the relation between time and brain-blood ratios. That gives us the rate at which the substance is crossing into the brain, the Ki, or unidirectional influx constant. And here's some actual data. What we've done here, we've radioactively labeled interleukin-1 alpha, and we've injected that into the bloodstream of a mouse. We've actually co-injected radioactively labeled albumin, labeled with technetium, which we can distinguish from the iodine on the interleukin. So we're able to, at the same time, look at what's going on with albumin and interleukin-1 alpha in the same mouse. And as you can see, the line is essentially flat for albumin. The method is so sensitive, we can, if we go long enough, actually see the rate at which albumin is getting into the brain. And here we see that interleukin-1 alpha is rapidly entering the brain and eventually hitting a steady state. And so this is formal evidence that a cytokine is crossing the blood-brain barrier. We also did autoradiography. We injected the radioactive ligand into the bloodstream, washed out on vascular space. So the only things remaining are those which have crossed the blood-brain barrier. And here we can see a capillary and the interleukin-1 dispersing out from the brain. Cytokines are around 16,000, 17,000 Daltons, really large, which means albumin is a good control for them because albumin is only about 4 times larger. But the only way for a substance really to get in that quickly that's this large is probably by a satchel transport system. And here we've formally shown that. Here's the experiment I just showed you, radioactive interleukin-1 getting into the brain. Now we've repeated that, but included unlabeled interleukin-1. And you see that the rate of entry has gone down, inhibition, ergo, satchel transport system. So we can do this for a number of cytokines, just kind of work through them and see which ones are transported and which ones are not. And besides self-inhibition, we can also do cross-inhibition. So we can ask, for example, is interleukin-1 alpha inhibited by other interleukin-1s? And in this case, we can see that they are. So we can conclude that they're all sharing a family of transport systems. And then we can ask, is TNF crossing the blood-brain barrier? And we see that it is. We see that it's a self-inhibitable system. But TNF doesn't inhibit interleukin-1 entry, nor does interleukin-1 inhibit TNF entry. So we know those transporters are separate. So by playing these games of inhibition and cross-inhibition, we're able to assign cytokines to specific families of transport. So I want to move on now to secretion of neuroimmune substances by the blood-brain barrier. And these fall into sort of categories. There can be a constitutive secretion, which brain endothelial cells do. And there can be a stimulated secretion. And then there's a very interesting thing. Remember, the blood-brain barrier, the brain endothelial cell is polarized. In other words, there's a brain side and a blood side, and they're separated by the tight junctions so that the contents, the lipids, the regulatory proteins, the transporters that are on the luminal side are not necessarily the same that are on the abluminal side. They're two different universes. So interestingly, you can have a situation where the brain endothelial cell receives an immune stimulus on one side, but then releases something on the other side. And this is a true axis because it's transmuting information from one side to the other. And we have a very common example of that, and that's fever. Again, the idea is that a neuroimmune agent is acting on one side, it's inducing a secretion that then acts on the other side. And what we have is a great example is interleukin-1, or actually almost any neuroimmune substance that produces fever, interacts with the luminal side of the blood-brain barrier, releases prostaglandins into the abluminal side of the brain. And this is what mediates fever. And this is why non-steroidal anti-inflammatory drugs are so good. But the source of prostaglandin is the brain endothelial cell. So we were interested to just show you one type of experiment that we do in looking at cytokine release by brain endothelial cells. So a neat trick is if you take capillaries and you plate them on a plate, they will stretch out and they'll become monolayers, and they will keep their orientation so that the abluminal or brain side is down at the bottom and the luminal side sticks up into the solution. And we have transwells so that we can sample either the luminal surface or the abluminal surface. So we've done that. We've plated some brain endothelial cells, imagine, with abluminal and luminal surfaces. And we're inventorying what cytokines are being constitutively expressed. And we found here these four: GCSF, KC, MCP1, and RANTES. We then added lipopolysaccharide, a component of bacterial cell wall that activates the innate immune system. And then the endothelial cell secretes a whole bunch of other cytokines. And here's a visualization for one of those, IL-6, no constitutive secretion, but with LPS, there's measurable secretion, both into the luminal and to the abluminal compartment. We then tricultured these with pericytes and astrocytes to see what that would do, how that would modify the brain endothelial cell secretions. And I'm sure you've all heard about astrocytes, but pericytes are still sort of the relatively new kid on the block. So I'll talk a little bit, just introduce them to you. They're the most intimately associated cell with the brain endothelial cells. In fact, if you can imagine a glue going over everything you see here, the basement membrane actually encapsulates and covers the pericyte and the endothelial cell, whereas the astrocyte is outside of that basement membrane. They're actually connected by peg and socket apparati that sort of physically puts them together. They're also connected by tight junctions, so they actually can share cytoplasm. So pericyte's the first cell embryologically that tells the blood-brain barrier to become a blood-brain barrier. Pericyte's also a stem cell, and it secretes cytokines. So here's sort of a summary of what we found. Again, if we have brain endothelial cells only, I've shown you this data. No LPS, we get 4 cytokines. With LPS, we get 12. If we now try culture brain endothelial cells with pericytes and astrocytes, constitutively, there's now 10 cytokines being released. And with LPS, it goes to 14. Here's the cytokines that I've shown you before that were induced by the endothelial cell with LPS. Here's what happens if we have a triculture. You see they're overlapping, but not totally identical groups. And then if we add LPS, we get still more cytokines. So what we think is going on is that some of this is crosstalk. We went on to do one more complication of this experiment. We incubated mouse endothelial cells with human pericytes and astrocytes. And then by doing differential assays that would only detect cytokines that were human origin or mouse origin, we're able actually to piece out which of these various cytokines came from endothelial cells and which came from pericytes and astrocytes. But the main point I want to make now is that by combining the cells, we're able to get even more cytokines, which is evidence of crosstalk amongst them. So the conclusions of this body of work was that brain endothelial cells secrete cytokines, both constitutively and with LPS secretion. And they secrete both interluminal and abluminal components, which is important if we're thinking of axes. And pericytes and astrocytes also release cytokines, some of which induce the endothelial cells to produce still more cytokines. And this is evidence of crosstalk amongst the endothelial cells, pericytes, and astrocytes. So I want to talk just briefly about modulation of barrier functions. This is conceptually the most complicated of the 5 areas which the blood-brain barrier and your immune system interact. And I'm not going to go through all of these. I'll just point out that there's 2 broad categories. An immune substance can act directly on the blood-brain barrier, or it can act indirectly on a peripheral cell, inducing it to secrete something that then acts indirectly on the blood-brain barrier. And an example of this is a regulation, neuroimmune regulation of insulin transport across the blood-brain barrier, a very hot topic in AD these days. And that's controlled by nitric oxide, but not by nitric oxide from the endothelial cell, but from nitric oxide from a peripheral circulating cell. So now I want to talk about disease conditions, states, and physiological aspects that are modulated or regulated by these systems. And the first one is sickness behavior. This is something most of your patients have, and certainly all of we have had. If you've had a viral infection, hopefully not COVID, or a cold or something like that, you've had sickness behavior. This was defined by Dancer about 20 or 30 years ago. And he said, you know, when you have these conditions, you get lethargy and sleepiness, depression, you lose your appetite, you're anxious, you're irritable, whole lots of things going on. And this is actually a syndrome. Later, he said, and you know what, this is an adaptive change in motivational state of the organism that reorganizes the priorities in order to cope with infection. And the first part I thought was brilliant that he realized this, but the last part is actually something that I think all our grandmothers told you, that when you get a cold, you rest so you can fight off the infection. But we were interested in how our cognitive changes induced through this. And we had the idea that cytokines crossing the blood-brain barrier were responsible for this. And the reason is because a great deal is known about sickness behavior at this point already. Classic sickness behavior is mediated by one cytokine, interleukin-1. Alpha and beta, either one can do it. Other cytokines can induce it, but the common pathway, the pure pathway, is interleukin-1. And it's long been known that if you inject interleukin-1 into a mouse peripherally, you can block many of the effects, including that of memory, if you put a blocking antibody into the central nervous system. So we found this interesting because we knew that interleukin-1 not only crossed the blood-brain barrier, but it had a big hotspot right here in the posterior division of the septum. That was interesting because the posterior division of the septum is this relay on the royal road from the hippocampus and subiculum down to the midbrain that mediates many of cytokine-like effects, including the sickness-like behaviors. So we postulated that this might be where interleukin-1 is coming in. If we could block that, we might be able to reverse the cognitive impairment. Now, as I say, it was already known that if you inject interleukin-1 into the bloodstream, you can block the cognitive impairment by putting a blocking antibody into the central nervous system. But no one believes cytokines could cross the blood-brain barrier. So they said, oh, it's a relay system. It's like going through the vagus and releasing cytokines from the brain, or it's acting at the endothelial cell, making it release things, or immune cells across, and all of these things. But it's not the cytokine that's injected. So after thinking about this for about 10 years, we realized that there was the perfect experiment was to take a mouse and inject inhuman interleukin-1, and then see if we could block the effect, not just with a blocking antibody to interleukin-1, but with a blocking antibody that was specific for human interleukin-1. Because in all of the relay mechanisms, even though you might inject interleukin-1 that would be human, what would actually be working in the brain would be mouse interleukin-1, only in the condition where the active ingredient is the interleukin-1 that you injected, crossing the blood-brain barrier, and therefore human working at the site of action would require blood-brain barrier transport. So long story short, we did that. I wouldn't be telling a story otherwise, right? We put in interleukin-1 into the antibody, injected into the antibody, and it changed the transport. It changed the cognition. So it turns out the blood-brain barrier is involved in a lot of sickness behavior. This is the one I just told you about. Interleukin-1 crosses the blood-brain barrier, acts at the PDS to affect cognition. We've also already talked about this one. That is through COX-2 prostaglandin release that mediates fever, but it also mediates changes in the HPA axis and through the striatum malaise and discomfort. Some people think that anorexia is involved in this system, a similar system too. And this one's also been worked out by Dancer, and we published one paper with him on this as well, but mostly it's a lot of work by him, showing that an inflammatory agent like interleukin-1 acts indirectly. Remember, we talked about that as being one pathway, releasing interferon gamma and TNF that alters IDO, which stimulates tryptophan in the periphery being changed to kynurenine, which then crosses by the neutral amino acid system and mediates depression. There's a lot we still don't know about sickness behavior. We don't know how taste aversion or other sleepiness or irritability or immobility are going on. But a lot of blood-brain barrier, a lot of sickness behavior is mediated by blood-brain barrier. Switching gears a little bit, Barbara Stonestreet, who's a neonatologist at Brown, showed that if you, in an ischemic animal, she's using mostly fetal sheep here, that interleukin-1 crosses their blood-brain barrier as well. And if they're exposed to ischemia, that transport rate goes up. And you can block that transport of interleukin-1 if you give a blocking antibody into the circulation. So here's how Barbara explains that it's working under the normal animal. You have transport of interleukin-1 across the blood-brain barrier. If you have ischemia, that upregulates the transporters, and so more interleukin-1 is getting into the brain. If you give interleukin-1 antibody into the bloodstream, the interleukin binds to the antibody binds to interleukin-1. This big conglomeration is unable to be transported across, so less interleukin-1 gets in. She then went on to show that this can change or can treat the sequelae of ischemia. So here you can see this is a normal blood-brain barrier, normal cortex of a fetal sheep. Here's one that's been exposed to ischemia, and you can see it's all whited out, a lot of damage. And then if you ischemia with the blocking antibody returns it to normal. So the interleukin-1 crossing the blood-brain barrier is responsible for this. Some more data. You can see that in the ischemic, the nuclei are shrunken and ptotic, and that are treated with the antibody, they're quite fine. The global pathological scores improve significantly as well with antibody treatment. Alan Butterfield at University of Kentucky has proposed this as an explanation for chemobrain. As many of you know, doxorubicin is notorious for causing chemobrain, but doxorubicin doesn't cross the blood-brain barrier because it's a substrate for P-glycoprotein. So how does doxorubicin do this? And Alan has shown that circulating doxorubicin in the bloodstream oxidizes ApoA1, which then interacts with macrophages to cause TNF secretion, which crosses the blood-brain barrier. And the TNF that has crossed the blood-brain barrier induces neuroapoptosis, resulting ultimately in chemobrain. Some of the TNF also stimulates the glial cells to release TNF from within the brain. So we get a double whammy here. And finally, a more recent example of cytokines crossing the blood-brain barrier and being important as a treatment. And that goes to the febrile infection-related epilepsy syndrome, FIRES, and another syndrome, NOVID. These are neonatal childhood diseases that induce seizures, and they're associated with high levels of interleukin-1 beta within the central nervous system. The seizures are by and large resistant to medications, including valproic acid, which is not a substrate for PGP, and to diet. But they respond very well to anakinra. So anakinra is essentially interleukin-1 RA receptor antagonist, which we showed many years ago, crosses the blood-brain barrier, plus one extra amino acid on the end that gives us a long half-life in the bloodstream. And so the idea is it's crossing the blood-brain barrier and thereby treating the FIRES and NOMID. Interestingly, the antibody directed against interleukin-1 beta is not very effective at treating these syndromes. And again, that makes sense from a blood-brain barrier point of view, because antibodies just don't get into the brain as well as the cytokines. Remember the first figure I showed you with albumin, antibodies tend to accumulate in the brain even less well than the antibody. So now I'm going to talk about some of the clinical applications of secretion of neuroimmune substances by the blood-brain barrier and modulation of blood-brain barrier functions sort of together. Because if you think about it, this is kind of the same side, or two sides of the same coin, that on the one hand, the blood-brain barrier is secreting neuroimmune substances, and on the other hand, it's responding to them. So neuroinflammation changes a lot of things in the blood-brain barrier. The disruption, many influx transporters are altered, efflux transporters like P-glycoprotein, virus transport is increased, and immune cell trafficking is also increased. And the last of these two, PGP and LRP, we know are also impaired in Alzheimer's disease and are responsible for the brain to blood efflux of amyloid beta peptide. And so thought to be involved in AD as well as in drug delivery and epilepsy and sepsis. Conditions that disrupt the blood-brain barrier were sort of this is sort of the traditional view, traditional list: multiple sclerosis, of course, sepsis and stroke. But it's been added to this quite a number. Diabetes, Alzheimer's disease within the last 4 years has been clearly shown with imaging to have a disruption. Chronic pain syndrome, at least in animal models, and even with healthy aging, there's evidence. And I hate to say this because I'm over 60, that people over 60 seem to have a small leak in the hippocampus. Whew, that was hard to say. And with the advent of the microbiome, we're increasingly interested not only in what lipopolysaccharide does in cases of disease states, but even if it might play a role physiologically. So here's an interesting example of how some of these elements fit together: cerebral cavernosus malformations, hemiangiomas of the brain, and clinical symptoms or seizures and focal deficits, neurological deficits and strokes. And this arises because of a loss of function mutation in Notch and MAP-ERK and GGF beta, resulting in pericyte loss. And you may remember that pericyte loss, pericytes are the first cell to tell the blood-brain barrier to form a blood-brain barrier. And their loss has been implicated in blood-brain barrier disruption in other diseases as well, including Alzheimer's and diabetic retinopathy and diabetic blood-brain barrier disruption. So that's what's thought to go on here. However, this is not constitutively expressed. It has to be activated by binding to TLR4 on the brain endothelial cell. And a recent paper about 4 years ago has shown that the source of that is likely the microbiome. And you can have a good microbiome or a bad microbiome for activating this condition. Alzheimer's disease has been involved with neuroinflammation and blood-brain barrier issues now for about 20 years, and in all sorts of different ways. Neuroinflammation in AD brain, there's increased inflammatory mediators, microglia are known to be activated. LPS is known to regulate the A beta influx and efflux in animal models. Infection and inflammation is known clinically to worsen or unmask AD. Inflammatory genes are associated with increased risk of AD. And the one that I don't think survived too well, but was very popular about 10 years ago, was that some nonsteroidals may reduce the risk of AD or AD progression. So there's all these different ways in which AD and inflammation have been sort of put together. And in terms of AD and the blood-brain barrier, one of my hobbies is to collect ways in which the blood-brain barrier has been associated with AD. And I'm up to about 15 now. And some of these are more popular than others. A decreased CSF reabsorption results in accumulation of neurotoxins. Defective glucose transporters, that's a very popular one. But one that, again, has sort of been reborn by the work of Zlotkovic is a leaky blood-brain barrier allows toxins to enter the CNS. Here are all the things that inflammation does to the blood-brain barrier that are also altered in AD. And disruption, decreased tight junctions, altered glucose transport, A beta efflux, et cetera, pericyte loss, all can be induced by inflammation and are occur in AD. Here's old data that was often thought to be evidence for disruption of the blood-brain barrier in Alzheimer's, or at least in normal aging. And that's the CSF serum albumin ratio. And you see it both sort of supports and denies this as a phenomenon. Clear regression of increase between the ages of 20 and 80, but you can see there's a lot of variation, so that the oldest patient has a level that's about the same as the average 20-year-old. And it's also, there's another explanation for this, and that is with aging, there's decreased reabsorption of cerebrospinal fluid, and that can also result in increased CSF serum ratio of albumin. So it was not until imaging came along that this debate was really resolved. And as I say, most of that work has been done by Zlotkovic, but just last year, Verhagen produced 2 papers that really sort of gilded the lily, and I think convinced a lot of people that were still not quite on the fence. And here is blood-brain barrier leakage as measured by DCI MRI over folks from about the age of 45 to beyond 90. And you see there is indeed a progression, but much as it was for albumin ratios, we have the oldest patient has one of the lowest levels. So there's a lot of hope here for individuals. So, you know, as a group, getting older is not good for your blood-brain barrier, but as an individual, you might be okay. And they also showed in a follow-up paper that leakage correlated in patients with MCI with decline in a very few cognitive tests, one being decline in delayed recall, and either the leak in white matter or gray matter correlated with that. But a very similar issue. Overall trend for the group, highly significant, but with lots of hope for aging individuals. Disruption of the blood-brain barrier by inflammation is not that easy. Here we've given LPS into animals, and this is weight loss. And you can see a very little, the lowest dose produced weight loss. But it took the highest dose to disrupt the blood-brain barrier. So the blood-brain barrier is rather resilient from this point of view. And we're now learning, or I guess we should say we're rediscovering, that blood-brain barrier disruption is probably not just because of tight junction dissolution, but also by reanimation of those other two ways that the blood-brain barrier is formed, the transcytotic and transcellular pathways as well. And here's what the blood-brain barrier looks like when it's high on inflammation. Here's ZO1, one of the tight junction proteins. And you can see they beautifully outline the cell. They're just on the periphery of the cell where the cells come together, cementing them together. And here is an LPS. You can see that the edges are ragged and thinner, and there's a lot of tight junction protein in the cytoplasm, thinking that what happens, it's not so much that the tight junctions go away, but there's translocation issues getting the tight junctions to the edge where they need to be. This is shows that with LPS treatment that the blood-brain barrier to sucrose is disrupted, and that indomethacin reverses that. But so that's a play on prostaglandins. But NAC, N-acetylcysteine, does not. So that says indomethacin, prostaglandins are involved. NAC, oxidative stress, doesn't seem to be. One of my other favorite hypotheses is the neurovascular hypothesis, which essentially says NAD, the brain-to-blood efflux of amyloid beta is impaired, and that allows accumulation of A beta and the progression of Alzheimer's disease. So I think we all know about AD plaques, and here's the AD molecule. And A beta is regulated, its entry and exit is regulated by the blood-brain barrier. In addition to enzymatic degradation, the brain also gets rid of A beta through clearance of with CSF reabsorption with brain-to-blood transport. And they're about 50-50 in regard to that. But there's also blood-to-brain transport by RAGE into the brain. And what this shows, measures A beta influx into the brain, and shows that it's increased by about over threefold in the face of inflammation. Using a method that we devised to look at brain-to-blood efflux, we found that efflux of A beta is impaired by inflammation. So inflammation, on the one hand, is increasing the rate at which circulating A beta gets into the brain, and on the other hand, it's decreasing the rate at which brain-produced A beta efflux is not being effluxed or getting out. So there's also an inflammation impairs CSF reabsorption. So, and it impairs the two components of the saturable efflux, LRP and PGP. So it's very interesting that inflammation recapitulates all these aspects of Alzheimer's disease with regards to LRP, PGP, CSF reabsorption, all of which affect A beta transport and accumulation, as well as that of other toxins in the brain. So I want to just go back and talk about one of those transporters for A beta, which is PGP. And PGP does a lot of things besides handle A beta, one of which is loperamide. So as physicians, you all know about loperamide because that's what we give kids and sometimes older patients when they have diarrhea. And the way that this works is that Imodium is an opiate, and it induces opiate constipation. We could just as effectively treat the condition with morphine. But morphine crosses the blood-brain barrier and causes mental clouding and other problems like that. And we usually don't want to do that just to treat diarrhea. And loperamide doesn't get into the brain because it's a PGP substrate. However, if you treat PGP knockout animals with loperamide, it's indistinguishable from morphine. You get the CNS symptoms. So this is a great example of sort of a very common aspect of how our blood-brain barrier is really nice to have around with all of its efflux systems. Another one is caffeine. If you like to drink coffee, this is my favorite story about the blood-brain barrier. Caffeine is transported into the blood-brain barrier by an adenosine. And I'm absolutely sure there must be a family out there that has the orphan disease of an adenosine transporter that doesn't recognize caffeine. And for those poor people, you know, every cup of coffee is decaffeinated. And I think that would be a horrible way to live. So lots of things transported by PGP. I think around 1/3 of the pharmacopoeia are PGP substrates. And a lot of clinical stuff can be explained from this. And just to touch on one, for example, in inflammation upregulates PGP that transports out more of the anti-epileptic drugs. And this may be the mechanism by which status epilepticus and its resistance to drugs occurs. So I think I just have enough time to whiz through some of our work on SARS-CoV crossing the blood-brain barrier. The fear is that because of the cytokine storm, this is the underlying problem with long COVID, and that actually this could even lead to long-term dementia. The viral attachment protein, the S1 protein, tells the virus where to go. So we have done, looked at, can S1 cross the blood-brain barrier, and can models of the virus, pseudovirus and virus-like particles also cross. Here's the data that was published just this year showing that S1 crosses the blood-brain barrier. You see it was very similar to what I showed you with the cytokines, and that we included albumin as a vascular marker. And S1 crosses the blood-brain barrier quite well. Interestingly, inflammation doesn't do much to increase blood-brain barrier transport of S1. If we were talking about HIV or GP120, inflammation greatly increases the transport of those. But other than helping with a little bit of the blood-brain barrier disruption, S1 doesn't seem to be facilitated by inflammation. It does, however, change the distribution and the uptake of S1 by other tissues. Here's one of my favorite slides, and I'll unpack it just a bit. So here what we've done is we've injected S1 directly into the brain, into the lateral ventricle. And if we take young CD1 mice, CD1 is the classic run-of-the-mill white mouse, we only see 2 cytokines that are increased with the dose that we gave, IL-12, P40, and RANTES. However, if we take an Alzheimer's disease mouse model, the SAM-P8, same age as the CD1, we get 4 cytokines that we actually get more, but I'm just showing these 4. We get 4 cytokines that are increased here, MIP alpha, beta, again, RANTES, and IL-12. If we now let those SAM-P8 mice age, and I should say when they're 2 months old, they're really indistinguishable from CD1 in almost any measure, including cognitive measures. By the time they're 12 months, they have profound cognitive impairment. And now when we give them give them S1, we see not only do the cytokines increase, but they increase more so than they do either in young SAMs or in CD1s. So what we're concluding from this is that S1 in the brain, once it's there, can cause neuroinflammation. It will do so more so in those animals that are predisposed to AD. And it does so even more to those who either have functional AD or had aging. We're devising experiments now to sort of parse out how much of the robust effect of the old SAM is due to aging and how much is due to activation of the AD phenotype. Likewise, if we incubate S1 with brain microvessels, it doesn't really do much to the tight junction cloud in 5, but it decreases by about 20% GLUT1 expression. And GLUT1 is, of course, decreased in AD, decreased by inflammation. And if SARS-CoV is also able to decrease GLUT1 transport across the blood-brain barrier, that's not good news. Here's an early study, with the SARS pseudovirus. I just got data from the BLP yesterday and haven't had time to work it up into a slide. But this shows that the SARS pseudovirus is also crossing the blood-brain barrier. If we inject that into the brain, here's control, here's virus injected. It has a GFP label, so we can see that, which is stained green, so we can see the virus is here because it's green. But it also activates, as measured by IABA, microglia, and astrocytes as measured by GFAP. So the virus itself is also causing neuroinflammation once in the brain. So that's my talk. I hope I have a few minutes for questions, but just quickly, the blood-brain barrier and the neuroimmune system interact in ways that we've divided up into neuroimmune axes in order to make fun and games more palpable and more easy to study, and that those interactions are producing a lot of the basis of a lot of disease processes that we see in clinics. Thank you very much. Yeah.
Thank you, Bill. That was a lot of, that was a whirlwind. That was amazing. I know people have a lot of questions. A couple popped up
Are
in
we
the
supposed
chat.
to raise our hands?
Yeah, sure.
Whoops. I have many reactions. Oh, there we go. Raise hand.
Oh, well, yeah, go ahead, Simi.
That was just fantastic. Thank you so much, Bill.
Thank you.
That was really cool. So I was wondering earlier, it was neat to see how the LPS model plays out in all the different mechanisms. Do you think that there's are there intrinsic molecules that are initiating those same inflammatory pathways that are a result of aging, or something that, your LPS is a model for turning it on, the inflammatory process, or are there other molecules that do the same thing?
Yeah, there are other molecules that do the same thing. LPS is readily available and cheap and extremely powerful.
Yeah.
So it's a great tool. But the problem is it's powerful. And so it's, like blasting the system. And there certainly, and we've looked at which cytokines it releases. Look at a 23 cytokine panel, it will increase about 14 or 15 of those sequentially. So one big question is it really stimulating all 14 directly, or is it stimulating 2 or 3, and then those are cascading? And the immune system loves to do this, right? Loves to cascade and positive feedback and all this great stuff. And that's still not really been worked out, but it looks like what has been worked out is a lot of it's mediated, seems like direct release of IL-1, TNF, and probably IL-6. So those are mediating a lot of those things like that. And the other interesting thing is that LPS, inflammation in general has gone from, okay, this explains diseases, to, oh no, these systems are also involved in normal physiology. And with a great interest in the microbiome, I think the experiment that is left to be done, it looks like there's LPS in natural circulation, in the absence of disease. And there's certainly LPS when you do certain normal things, like if you jog real hard, or even if you eat a high-fat meal, LPS comes in. Why? I don't know. But I suspect that there will eventually be a physiological level of LPS, and that we're dependent on our microbiome for that as well.
Oh, that's so neat.
Tune back in 10 or 15 years.
Yeah, that's cool. Thank you.
Should I run the chat or do you want to?
Yeah, I can read it for you. So,
Okay.
yeah, Mike Kim asked about post-acute sequelae of COVID-19. Is there evidence for increase in IL-1 or IL-6 in these patients?
I'm sorry. Well, oh, I see.
Yeah,
Yeah, so there have been cytokines measured in the cytokine storm. There are, I'm trying to call, there are about 18 that have been measured. It's like a lot of early clinical stuff, you know, it's a little bit piecemeal. So this group over here measured 4 cytokines that are all elevated. No one's quite gotten around to using multiplex technology yet. But there's about I think about 14 or 18 in the literature now. A lot of them are sky high. And I hesitate to mention any because I'm sure I would get them wrong. I think IL-1 and IL-6 were there. Not sure about TNF. And I think that what surprised me was I think the GM-CSF was very high. The GCSF. So some, I guess you would call those chemokines as well. So, and in terms of interest to us, and I should know this better because I did put it in a grant, but there's been another grant between this one and that one, that there was a nice overlap between what was seen in the cytokine storm of COVID and what you get with LPS. Not perfect, but there was some redundancy.
Mm-hmm. Andy is raising his hand.
Hi, thanks. Hi, Bill. Thanks for the great talk.
Hi, Andy.
Hi, how are you?
Fine.
Yeah, so much history that you've given us, and so many amazing things to look forward to in the future. My question is about the receptor for S1, and there was this idea that maybe parasites were one of the targets, even though they're on the abluminal side of the endothelium,
Right.
but they express more of ACE2 than the endothelium does. And I was curious about whether or not your studies had looked at the impact of the S1 protein on parasite, I don't know, viability, function, adherence, and whether there's interest in looking at this in vivo.
We have not yet, but you know, that kind of question wants me to leave the lecture and go back to the lab because that's really a great question. My lab claims to be the first to show that pericytes secrete cytokines, and I love the pericyte. It's after the brain endothelial cell. It's my favorite cell. So I think that's a great question and needs addressing. I thought you were going to ask me about ACE2. And our studies that we did indicate that ACE2 is important for lung and maybe for brain, but not for some other tissues. And I think we've all gotten so excited about ACE2, we've forgotten that viral binding and viral protein binding is not Michaelis-Menten. It's much more lectin-like and promiscuous. And, you know, there's a lot of viruses that will bind to 4 or 5 main receptors. So I suspect that ACE2 will not be the whole story here. And I think there will probably be other receptors or other binding sites that the virus takes advantage of.
Awesome. Thank you.
Thank you. Yeah.
While we're talking about parasites, I was wondering if there's any evidence of maybe some somatic mutations that could arise in parasites in aging that might explain some of the aging phenotypes. Or
yeah, one area I've read a little bit about that I think is really intriguing is perhaps you guys have read more about somatic mutations, which, you know, occur apparently at the highest tissue that has this, the central nervous system. Mm-hmm. And I just think this might explain a lot of stuff. But I haven't gotten around to, you know, doing anything with that. And, you know, the main reason, of course, to say, yeah, I give this talk here is hopefully to stimulate collaborations, which are ongoing with some of you at a slow pace, but hopefully we can get ideas and speed up. There's just so much to do, you know. So I don't know, but yeah, I think pericytes seem to be really, even more so than astrocytes or microglia, central to blood-brain barrier diseases, particularly those related to disruption right now. So really interesting.
No, Jerome, did you still have a question? Or
Yeah, I put it in the chat.
oh, sorry.
I love everything about this talk. Thanks so much, Dr. Banks.
Thank you.
I was going to ask, we see a lot of CAR T neurotoxicity here with the Cancer Center and their patients. So,
Yeah.
and there's been some work on it, you know, from Cameron Turtle looking at cytokines, but there's kind of this debate in the literature about some of it seems to be cytokine release, blood-brain barrier, maybe dysfunction, versus this other theory that says some of it is this cortical excitotoxicity. Has your group kind of looked at this? Because we see a lot of it here. CAR T use is expanding rapidly.
Yeah.
And now people are using like CAR NK cells and different ones. And a lot of the immunology we just don't know that much about. But so far, all we're using is steroids and tocilizumab, and they seem to help. But it seems like your work might make us better at preventing it or treating it better, because sometimes the outcomes are really extreme.
Yeah, I mean, as far as I know, in most cases, you're not really wanting a CNS effect, and so if that could be blocked or treated, that would be wonderful. But no, we haven't done anything. We just don't have the models. If we have the models around collaboration or something, I think it'd be a piece of cake to at least look at disruption in an animal model or something like that. In theory, you could also, I don't know if anyone's looked in humans with the syndrome to see if the blood-brain barrier is disrupted or not. But there's pretty good methods for that now. So.
Oh yeah, and I guess I've missed two other things in the chat box from Seth Perlman. He says, I was struck by the concept of using anakinra for fires. It's been a while since I saw a case. It's very devastating. It sounds like quite a recent development. Do you have a sense for how effective and common this treatment approach may be?
It looks extremely effective, at least compared to anything else. I recently sort of reread the literature, which used to not be difficult because there were like 2 papers. And now I think it's ballooned to like 8 or 9. And so there's actually now some cases where anakinra was less than 100% effective. And I think there's one report where there was an anakinra was less than effective and they found something else that worked, although I don't recall what that was. But there's always been the issue from the very early days, how much to give and how long to give it. Because, these, I see the chat says, it's really devastating. I mean, it's just hard to believe. I've never seen a patient with this. So I've just read the clinical studies, and it just seems hard to believe how horrible this is, that children have like 100 seizures a day and things like that. Almost status epilepticus, I guess. But anyway, the original studies found it very effective, but it seemed that they had to maintain people on solutions, kids on IV solutions for the longest time. Some of the follow-up seems to say that you can wean them off. There seems to be better ideas of dosing now, but I think it's very much an area that needs a lot of study and research.
Yeah, and there are a couple more new comments. And it's being used at children's, not sure when to wean it off. And then Sung Ji has a comment. Thank you, Dr. Banks. Has any have any studies looked at whether there's more incidence of COVID-19 virus seen in the CSF with patients with underlying conditions that disrupt the blood-brain barrier compared to those without clear disruptions?
Yeah, I don't think that there's been studies really with COVID-19 and blood-brain barrier disruption. There's been, from the earliest days, evidence of encephalopathy, like the first Chinese cases reported. But I don't think that anyone's actually studied blood-brain barrier clinically, or even other than us. Well, us and a few other papers, even in mice. I think, let's see, I'm looking for, I think I've missed the essence of the question though.
From Sung Ji.
Okay, yeah. So there, and again, there hasn't been that much viral recovery. I mean, there haven't been that many studies published that have looked at it. And whether, so I've been focusing more and more on sort of the long COVID question. One study found, and there's been two studies recently, both out of England, that really sort of finally give some light on this. One was that people that were the sickest of the COVID-19 patients, the one on respirators, had what was equivalent of a 7-point drop in their IQ. I think it was eight weeks after. Of course, there's there's the respirator effect. So I don't know how much that is. But the most frightening one came from UK just about 4 weeks ago. And they found, this was self-reporting, but they had like 3,000 cases from all over the world. And they found that people who had long COVID, the majority were not hospitalized. They weren't that sick, but their symptoms never went away. And at 7 months, the number of patients that were asymptomatic were still only about 5%, hadn't increased over time. So it really was long. And they had shifted from very severe and severe to severe and moderate. And that was all the progress that they made. The average patient had 56 symptoms. And at the top of the list were brain fog and that kind of thing. And things like fatigue, which I think would probably not be muscular, but central nervous system mediated fatigue. So anyway, so all that's very interesting, I think. And of course, all this is, people have cleared the virus. So this isn't S1 lingering. I think this is neuroinflammation. This, the brain is very upset, neuroinflammation gone wild.
I had one other question, I was wondering if you had any thoughts about transport of extracellular vesicles or exosomes from the brain to externally or vice versa.
Yeah.
If there's if you think there's active transport of those types of vesicles, or if it's, I don't know, through, I was just wondering if you.
Yeah, that's really interesting question. We did a little bit of work with Jay-Z, who used to be here and unfortunately left. And he was one of the original guys to show exosomes from the brain getting into the blood. And we did a study where we looked at, I think it was Parkinson's vesicles in the blood. And we did one study and we found that our interpretation, we could inject vesicles into the brain and they would pick up alpha-synuclein and transport it out. So we think there's some packaging going on rather than just dependent on tissue secretion. And most of our work in the exosomes, though, has been in the blood-brain direction now that Jay-Z's left. And it's a little bit easier model to do than brain to blood. But I think that's really a cool area that exosomes can get, CNS origin, and it's just filled with all kinds of potential.
Yeah. Well, the other way around too, for packaging drugs or something. Yeah, great. Well, I don't know if anyone has any other questions before we enjoy the last bits of sunshine.
Yeah.
Yeah. Thank you so much. That was a great talk.
Great. Thank you all so much. Take care.
Thank you.